Yep. So my name is Jason Whitham, and today I'm going to be presenting to you how to use FastANI and Mash for rapid whole genome comparison and how I used it to find a novel myxobacteria genome in a metagenome data set. Pictured above, the wolf is an example of a slimy myxobacteria. Myxobacteria are really cool for many reasons. One reason is because they are predators that work together to eat other microbes. Microbiologists actually use the term wolf pack to describe the cooperative killing. Shameless plug here. This is a great teaching narrative, so please don't steal it. Just ask me and I will be happy to be a guest lecturer or a co-instructor of your course. I need the teaching experience and I am cheap labor, as Carlos will testify. So what am I going to learn today? Throughout the seminar, I will ask questions from your perspective. We're going to answer this question by asking and answering these other questions. What is ANI? How are genome comparison metrics useful? What software packages are available to calculate these metrics? How does FastANI and Mash work? What databases are available to query? And what are some test commands that I can repurpose for my research? We're also going to do a bonus. We're going to do a little demo of Mash at the end. If you have access to Henry 2 and have the files I shared, you can do the demo along with me. If not, you can just watch me do it. So what is ANI? It's an acronym for Average Nucleotide Identity. The way it is calculated is a genome is chopped up into fragments. The fragments are aligned to a reference sequence. The best scoring matches are saved. The same procedure is repeated with the reference. Then the identity scores are averaged. You expect high scores from genomes that are similar, such as in the case of genomes from different Pseudomonas putida strains depicted here on the right. So what can I use it for? You can use it for comparison and identification of organisms. Alternatively, you can compare organisms by universal markers, but probably only to the genus level, if that. You can get to the species and strain level with ANI. The ANI quantity is more meaningful than a semi-quantitative value from Southern blot, and it's more accurate than the average amino acid identity because amino acids can be translated from multiple codons. But it does take longer to calculate ANI than AAI. You can also search databases with a genome to find out if it's novel, or find a complete or more complete genome from a partial genome. These are just some common applications. So what software packages are available for rapidly comparing genomes with ANI or something like ANI? There are several. I picked a couple of high utility ones to test on Henry 2. Both calculate ANI-like metrics. To save computational resources, k-mer sequences are converted to hashes. That's common between FastANI and Mash. One of the major differences in the approaches is that FastANI only uses shared genomic regions, while the Mash metric is penalized by differences in query and reference sizes. So what's the advantages of considering shared regions? When FastANI performs a bidirectional search, regions that are most common are saved. Paralogs, which are homologs on the same genome, do not inflate the FastANI values. Only orthologs, homologs in different species, which are more similar than paralogs, are used for FastANI calculation. So what's the advantage of considering all regions? Here I'm showing an unpublished figure that I made. It's an alignment of phage reads on a bacterial genome. The abundance of phage reads increases relative to bacterial genome reads as the retention time of fermentation reactors decreased. I used this here because I couldn't find any good figures online of what it looks like to have bacterial genomes containing phage. The Mash calculation penalizes differences in reference and query size to avoid getting matches of phage with a bacterial genome containing a phage. Any other calculation differences? Yes, FastANI uses the Jacquard index or intersection over union of hashes, whereas MASH uses an estimation of the index by incorporating N, the average number of hashes for 2 genomes. Look at the bottom right corner of the slide. W is the total number of hashes for 2 genomes being compared. So if the 2 genomes were the same, N and W both could be 100. J could then be 100 divided by 2 times 100 minus 100, which is equal to 1. And the natural log of 1 equals 0. So the natural log of anything less than 1 will be negative. So the negative sign makes the final MASH value positive. The K refers to the chosen size of Kmer. The default is 21. And FastANI does a similar calculation but uses log base 10 and adds 1. So queries and references that are similar will thus have a high FastANI close to 1 or low MASH distance close to 0. There's more to it than that, but for the sake of time, I ask you to check out the references for more details on the math. Now let's get into some test results. I did some tests of FastANI and MASH on Henry 2. Rather than searching for my myxobacteria in a really large database right off the bat, I started with a smaller curated database called RefSoil, just 922 soil organisms. Unfortunately, the first time I tried querying the RefSoil database, I found no result. I should have tested a control first. When I queried a genome from the RefSoil database against the RefSoil database, it still didn't work. I discovered that I needed to decrease the fragment length parameter. That makes the query fragments shorter, more hash hits. With higher sensitivity to others, lower specificity, and longer runtime. So what is the optimal frag length? After 3,000 base pair, I tested 1,000 base pair and got way too many genomes, 493. So I tried 2,000 base pair and again got no genomes. Then I tried 1,500 base pair and got 4. 1,200 base pair got me 25, which seemed like a Goldilocks number. Wait, did FastANI match similar genomes to query when you used a fragment length of 1,200 base pair, Jason? Yes, and here are the actual hits I got from RefSoil database. I selected NC015733 from the RefSoil database for my query genome, and it's the top hit. I don't know why the bidirectional frag mappings are not equal to the total query frags, but they are not. NC015733 is Pseudomonas putida strain S16. For those who care, Pseudomonas putida is a common soil bacteria that has many metabolic pathways for breaking down a large variety of compounds and is consequently studied for biotechnology applications. It was actually the first bacteria to be patented. The FastANI publication compared 8 billion genomes, calculating ANI values, and found that ANI could distinguish species. They found that 99.8% of the time, species had an ANI of 83% or higher. This does not mean that we won't see other species that are above 83% ANI. It just means we shouldn't see other Pseudomonas putida strains lower than 83%. The good news is that the lowest ANI for Pseudomonas putida strain in this example is about 87%. Notice we do see some non-putida species that have a higher ANI value than the W619. Pseudomonas putida strain near the bottom. Hopefully that's okay with you. It's okay for my purposes. Now that we have optimized our fragment length for searching the RefSoil database, let's see if we can find my myxobacteria mag. The closest hit is Syringium cellulosum. This bacteria is a myxobacteria. A picture of it is shown in the upper right corner. An old Wikipedia citation says it has the largest bacterial genome sequence to date. The ANI it shares with many myxobacteria is below 83%. So they are very unlikely to be the same species. What about partial genomes? Remember, it's important for FastANI to be able to utilize partial genomes because metagenome acquired from genomes are almost never 100% complete. So what happens if I query only a fraction of a genome, say half or a quarter? Does FastANI still find matches? It does. Remember how we said that FastANI only considers common regions between the query and the reference. That's why the lowest ANI doesn't change much for half and quarter genome searches, but our bidirectional fragment maps and total query frags decrease. We've talked about FastANI. Now what about MASH? How good is MASH at identifying similar genomes? Again, I use NC015733 to search the ref soil with MASH. MASH found our genome in the database with a perfect MASH distance of 0. You'll notice that the output of MASH is a little different than FastANI. Rather than giving us a bidirectional frag mappings value and a total query frags in separate columns, MASH gives us a Jacquard index value in one column. This is independent of genome length. Remember though, MASH penalizes in the Jacquard estimate, which ultimately impacts the MASH distance. So the p-value is dependent on the probability of the random k-mers appearing in genomes and the size of the sketch. We will talk more about what a sketch is shortly, but here you will notice the list of matching organisms found with MASH is similar to what we found with FastANI. So what is a MASH sketch? To save computational time, MASH generates sketch files of hashes from source files, queries, and references. It's possible to compute a MASH distance without making sketches, but making sketches saves a lot of resources if one plans to do multiple queries against the same reference. Sketch size is how many k-mers are converted to hashes per genome. For example, 1,000 k-mers, 10,000 k-mer size, or 100,000 k-mer size. So MASH can be fast, but is it less accurate than FastANI? According to the pioneering FastANI journal article, it is. They looked at 5 databases. FastANI had a very high correlation with BLASTANI, that's the ANI-B. MASH had low correlations with BLASTANI for some more fragmented genomes. They also showed that MASH performance improved with increasing sketch size. Sometimes 1,000 base per k-mers is not enough for some databases. Keep in mind that the larger the sketch, the more time it takes to make the sketch and perform the query. So we are able to query partial genomes with FastANI. Can I do it with MASH too? Even though it penalizes for genome size differences. To test this, again, I took a half and quarter portions of the NC015733 genome and queried against the ref soil database. Like FastANI, MASH was able to match the partial genomes to the complete genomes. Now that we've talked about FastANI and MASH, let's talk a little bit about databases. Ref soil is nice, but what about bigger databases? Should I use MIGA? MIGA is the Microbial Genomes Atlas. While the concept is great, that is, compare your query genome against many of the prominent databases, if you try this service, you will probably be waiting for a very long time to get output. What about local? Can we host a database on Henry 2? Yes, Lisa helped me get refseq 201 on Henry 2. Lisa is going to help me update it to version 204 soon enough, which has about 3,000 more organisms than our current version. In total, it contains 106,581 organisms. It's a lot of genomes. So how did FastANI and MASH perform with RefSeq? Sadly, FastANI is a memory pig. Using the 1200 frag length, after about 24 hours, my batch job quit. The maximum memory was over the Henry 2 limit of around 500 gigabytes of RAM. I proceeded to test FastANI with frag lengths of 3000, which is the recommended length, 9,000, so 3x the recommended, 18,000, so 6 times the recommended, and 30,000, which is 10 times the recommended. All failed, exceeding max memory except the 30,000, but it yielded no matches. So fragment length was too long. There is a way around this. And if you really want to use FastANI, I can show you how to do it. Basically, you have to break up your database into smaller pieces and then use the smaller frag length. So now we're going to demo and use MASH on Henry 2. So here we are on Henry 2. There we go. I'm going to go into my Scratch directory, and I'm going to change to my demo directory. And in here, we have NC 015733. Our search or query genome. And then we have our sketch of our RefSoil 201 database, and we have our script. And so to save a little time, I'm just going to go ahead and sub our script. See if it's running with bjobs. All right, it's running. And then we're going to nano into the script and see what it's doing. Okay. So this is a TCSH script. We selected something with 12 cores. We want exclusive use of the nodes. And we only need a wall clock time about 5 minutes. This is pretty fast. It should only take about 3 minutes. We need to activate our Conda environment where MASH is located. I've already made a sketch. That's what that first hashed out couple lines are. And what we're doing right now is we're estimating the pairwise mutation rate. That's the MASH value. It should be a low value. And here you can see that we're using the dist executable for MASH, and we're using 12 threads, and we're looking at the RefSeq database with our query genome NC 015773, and all of that is going to go to a tab delimited file called distances.tab. And then from there, what we're going to do is we're going to take this bottom command here. It's going to help us sort through and just display the top genome hits. So I'm just going to go ahead and copy that. I'm going to get out of my nano. Let's see how the job is running. Still going. So let's look at the B jobs again, still running. Shouldn't be much longer. Like I said, the last couple of times that I've tried it, it only took me about 3 minutes. It can go longer if you use less cores. It can go a little bit shorter if you use more cores. But depending on which one you ask for, you might not be able to get the resource and you might be in the queue for longer. There we go. So now I'm going to paste this command. All right, here we go. So this is a list. On the left side is different genomes that are in the RefSeq database. And they're labeled a little bit differently, but I can tell you that that first top one, the ASM 21970V1_genomic.fna.gz is Pseudomonas putida S16. Is the same thing as the NC 015733.fa. And I don't know why it doesn't have a perfect 1,000 out of 1,000 for the Jacquard index, but it's a match. And so that's what's important. So what about the myxobacteria? Was something similar in the RefSeq database? No, out of over 100,000 genomes, a couple of hits had Jacquard indices less than 3, translating to a very high MASH distance. So one hit is a CDS, which stands for coding sequence, so it's not a genome. The other hit is Rhodospirillus, is not even a myxobacteria, so that's exciting. The myxobacteria that I extracted from my metagenome appears to be novel based on these RefSeq genomes. The caveat is there are many other databases out there, so it could be included somewhere else. My conclusions are that Mash and FastANI are very effective at comparing genomes, checking a genome or MAG for novelty, and retrieving a complete genome with a partial genome. So we have Mash and FastANI on Henry 2. FastANI is more accurate, but Mash is faster and less memory intensive. The RefSeq database is available for query on Henry 2. You cannot use FastANI with a fragment length of 1,200, at least not without breaking up the RefSeq database into smaller portions to query, and just because of the memory limitation on Henry 2. And you may or may not have to use a higher fragment length if you break up the database. Here's my references. And I want to say that I greatly appreciate Lisa's help with getting FastANI, MASH, and RefSeq updates on Henry 2. She's really been great to work with. If you want assistance with this or other bioinformatic packages, the bug group may be able to help. Send me an email or try our listserv. Remember, please stay after the Q&A for a quick bug business meeting if you're interested in serving as a bug founder or leader, or email me if you can't stay for that. Thank you for listening. I hope you will join us for more talks in the future. We're targeting about 1 per semester for now. At this time, please feel free to ask any questions you have about the presentation or demo.